Thank you for tuning in to my talk today about recent advances we have made in predicting how functionalization and mutations affect stability. First, a very quick comment on protein structure. As you all know, protein structure is hierarchical. We start with the primary structure, which is the amino acid sequence, and then molecules self-aggregates or folds into secondary, tertiary, and higher order structures. The big concept to remember is that structure equals function. If this protein were structured any differently, it might become less effective and could even become inert. Harnessing the power of these proteins for specific tasks requires creation of protein devices. Protein devices consist of a protein that has been functionalized with molecules or structures different from the protein's native environment. Two common examples, which we study, are depicted here. The first functionalization strategy is to covalently tether the protein to a solid surface or substrate. This is commonly done to make sensors or detection assays. The second is the protein can be pegylated by attaching polyethylene glycol polymers to the biological molecule, such as used in pharmaceutical applications to slow filtration in the body, and many approved drugs use such a functionalization approach. A big problem when making protein devices is controlling where the functionalization takes place on the molecule. Traditionally, conjugations are limited to lysine and cysteine residues and the termini of the protein. This limitation creates a heterogeneous mixture of diminished functionality. Moreover, locations of the natural points of attachment may interfere with the protein function and render the device useless. Our work employs a state-of-the-art method to precisely control to which amino acid the functionalization occurs. Now, this is done by replacing one of the natural amino acids with a single unnatural amino acid at the desired location. This unnatural amino acid has a side chain whose chemistry is not found in nature, either an azide or an alkyne that can exclusively react to form a covalent tether between the protein and the device. What is unknown in this process is how the mutant unnatural amino acid affects protein structure and where to attach the device, because structure equals function and protein devices affect this structure. Predicting the consequence of mutation and functionalization is important. For this work, these two challenges are addressed using molecular dynamics simulation and asks the questions: 1. How do unnatural amino acid mutations affect protein structure? And 2. Where do we make these attachments? Done properly, protein devices can improve performance, functionality, and efficiency, but if done incorrectly, it could destroy protein activity. Until recently, it was impossible to simulate the unnatural amino acids used in our experimental process because model parameters did not exist for the linear chemistries of the azide and alkyne moieties in these unnatural amino acids. Earlier this year, we published an article solving this problem where we described charm-compatible parameters for these unnatural amino acids. One of the big questions we wanted to determine is if a mutation affects protein structure. Because we didn't have any predictive capabilities when designing a new protein device, both us and our experimental collaborators had always assumed that the mutations in protein devices were not a significant contributor to differences in stability and activity, and that any changes were from subsequent attachment to a surface or peg. However, recent experiments have shown this not to be the case. Here, we have T4 lysozyme, and these are the 4 locations where we have experimental mutant stability data. 4 different mutants were examined as listed on the abscissa. The ordinate shows the change in the melting temperature for the mutant compared to the wild type. A negative value means the mutant is less stable than the naturally occurring molecule. The data indicate that all mutations destabilized the protein, but that mutations at serine 44 and asparagine 53 were much more stable than mutations at histidine 31 or lysine 91. But what we want to do is know this information without doing the experiment. We want to know to avoid H31 and L91 and use S44 and N53 instead. We hypothesized that we could use simulation to identify problematic mutations And the approach we took uses all atom solvated MD simulations. We first equilibrate the wild type and mutants with NPT to get the right density, and then use NVT. After a production run, we would calculate the root mean squared fluctuation of each protein compared to the PDB structure and compare the RMSF of wild type to that of the mutants. We wanted to see if changes in RMSF were correlated to the experimental changes in melting temperature. We don't have time to examine all of the details of the structure changes from these mutants in this presentation, but let's look at H31 just a little bit more. H31 is located on the N-terminal domain of the protein and participates in an important salt bridge near the active site. Visually, we can see significant deviation from the wild-type structure and that this salt bridge breaks. T70 is the other participant and is located in the hinge bending region. Here, we also see significant deviations. This likely leads to decreased function and activity. To further examine exactly where the structure is affected, here we have RMSF plots where the protein is aligned to different domains. The important takeaway is that when we align to the region that includes the mutant, we get an overall poor fit. But when we align to a region that doesn't contain any of the salt bridge participants, then the fit is good for all residues except those close to the bridge. Thus, our MD simulations were able to identify even localized and fairly minor structural deviations lead to protein destabilization and a decreased melting temperature. So now that we have a method to identify how the mutation itself affects stability, we need to develop a process to decide how the functionalization itself affects the molecule. There were three main goals of this portion of the project. The first is to move beyond the quintessential model protein lysozyme to determine if we can predict behavior for a protein that is both industrially and medically relevant. We consequently chose TEM1. This protein is used in the purification of milk products and medicinally to stop allergic reactions to certain antibiotics. The other two aims seek to advance our understanding of folding behavior using an in silico screen. We want to compare and contrast well-performing sites using the two protein devices introduced previously. These are namely tethering and pegylation. And finally, with the results of our screen, we want to determine a rational design technique for identifying the best attachment sites for experimental testing for future applications. We screened or performed replica exchange on all 89 surface accessible sites on TEM1 that would not impede active site function. Depicted here are key results for tethering. The table has the numeric ranking of the best and worst performing sites. I've highlighted sites 153 and 4 as they perform well for both tethering and pegylation, as we will see shortly. The heat capacity curves reveal that no treatment improved upon wild-type stability, but there is a significant spread between sites that are most and least destabilizing. This spread is helpful from a screening perspective in that candidates for experiments should come from the blue population and not the red. Here are the results from the pegylation screen. We again see that sites 153 and 154 highlighted in green are good attachment locations. Notice from the heat capacity plots that the spread between good and bad sites is not as large as tethering, but separation still exists and it is sufficient based on our previous experience to adequately choose candidate sites. We examined many different primary and secondary structure factors in the hopes of finding a connection between these metrics and site performance. These include sequence number, amino acid type, secondary structure classification, and solvent accessible surface area. None of these factors clearly predicted device performance. This is important because many typical design heuristics are based on these factors. Our analysis shows that revised heuristics are needed. Since primary structure analysis didn't yield conclusive results, we will instead transition to an examination of tertiary domain analysis. Here, I've uniquely colored each domain in TEM1 for convenience. Colors in the protein's graphic correspond to the colors in the figure. For example, cyan is the alpha-1 domain, magenta is the beta domain, etc. The figure was taken from a wild-type only simulation and breaks down stability according to these domains. The metric plotted is the percent of contacts formed in the protein. Now, these contacts are the intermolecular phenomena such as hydrogen bonding between different residues, and these drive the protein to form the secondary and tertiary structure. Thus, percent contacts is a measure of the fraction of the protein folded, and the inflection point is used to identify the melting temperature for the domain indicated. Notice that domain stability is not uniform across the protein, and some domains denature before the overall protein melting temperature, and some remain stable even at higher temperatures. We use this information to rank the overall stability of each domain. For TEM1, the beta domain is the weakest and alpha-2 domain is the most stable. What is important to note is that this information can be determined from a simulation of the wild-type protein prior to simulation of the 178 functionalized variants. What we will show is that the wild-type behavior is a good surrogate to determine how the myriad functionalized proteins will behave. We did a similar domain analysis for the functionalized proteins. To visualize the performance, I'm going to show several slides with the 3D structure of the protein colored according to performance. Top performing sites are colored in green, followed by shades of lime to orange. The poorest performing sites are indicated in red. To help focus the discussion, the 2nd through 5th rows show specific domains. Let's first start with the beta domain. As indicated by the high concentrations of warm colors, namely reds, yellows, and oranges, this domain contains many poorly performing sites for both functionalization methods. The additional forces added to the system at the functionalization point on the device overwhelms the normal protein beta structure so that the protein melts at lower temperatures. Moreover, this instability propagates into other domains and results in a lower overall protein stability. If we recall, this beta domain was previously identified as the weakest domain in the wild-type analysis. This is significant because it shows that inherently weak domains, as determined by the wild-type analysis, will not produce many good sites upon functionalization. More work is needed to identify if it is beta sheets themselves that are poor domains to functionalize, or if this beta domain weakness is unique to TEM1. Let's now look at the alpha-2 domain. This domain is the most stable domain as identified by the wild-type analysis. It is the largest domain of the protein and contains many good sites for functionalization in tethered and pegylated models. It was previously believed that when functionalizing in structured domains, the additional forces the device produces on the protein structure would break the core structures and an overall loss of activity would result. While this was true for the inherently weak beta domain, this is not true for the more stable domains. This domain was resilient to the device and was often the last domain to denature despite the additional forces. The results here are promising because they show how the wild-type only analysis can identify good sites for further examination during the site selection process. Interestingly, this domain was not the domain with the largest number of well-performing sites for either tethering or pegylation. So in conclusion, we were able to successfully simulate unnatural amino acid mutation effects on structure and rank a protein device's effect on melting temperature in dependence on attachment site. The all-atom simulations disprove the previous theory that unnatural amino acid mutation effects are minor compared to the effect of the surface or peg. And by contrasting tethering and pegylation screens, we can conclude a device's dependence is more strongly dependent on domain location than primary sequence attributes. We are very grateful for the supercomputing resources at the BYU Office of Research Computing and our funding by the NSF. Without their support, none of this would have been possible. I'd also like to give special thanks to our wet lab collaborators working in the Bundy lab. It has been great presenting my research to you all. Thank you.